Hello, everybody, and welcome to the deep dive seminar on the lead article published in Frontiers in Science, Enabling Sustainable Crop Protection with Induced Resistance in Plants. I am Laure Sonnier. I am the executive editor of Frontiers in Science, and it's really my great pleasure to welcome all of you online today. In the Frontiers in Science lead article, Professor Brigitte Mauch-Mani and colleagues highlights the potential for broad-spectrum, long-lasting protection against pests and pathogens through so-called induced resistance by strengthening the plant's immune system through exposure to various stimuli. So the lead article and the complementary publications in this article hub outlines the next steps for the widespread adoption of induced resistance into integrated pest management strategies for sustainable crop protection. The article and its hub is a result of a collaboration between a large panel of worldwide experts, some of whom being with us today. So I'd like to extend a special thank you to Professor Mauch-Mani for your trust and for your leadership in bringing this important project to success, and to all contributors of the project for entrusting us and working with us to publish it in Frontiers in Science. We are delighted to have given it a home in our flagship journal. So just a few words about Frontiers in Science. We started conceptualizing the journal 5 years ago, and we launched it over less than 20 months ago. And it is our flagship multidisciplinary open access title at Frontiers. Our editorial scope focuses on transformational science to accelerate solutions for healthy lives on a healthy planet, and is guided by the United Nations Sustainable Development Agenda. So how do we work? We commission exceptional peer-reviewed lead articles from world-renowned researchers whose work addresses global challenges in human and planetary health. And then around each of these groundbreaking lead article, we built a unique hub of complementary content, including scientific viewpoints, editorials, policy articles, and also content for children and the general public. This is all built by design to expand reach and impact of this transformational lead article across scientific policy and public audiences. And really, to this day, no other publisher is doing this in the way we do it. And we are very proud to be driving innovation and impact in publishing so that critical transformational science can reach us all in society and accelerate scientific progress for every one of us. So today's discussion is going to be moderated by my colleague Rui Fernandes, who is one of our content editors at Frontiers in Science with a background in ecology. I'm going to now turn over to Rui, who will moderate the event. And I thank you again, all of you, for being with us today.
Thank you, Laure, and a warm welcome to everyone around the world. Good morning, good afternoon, and good evening, wherever you are. I'm delighted to be your moderator for this exciting deep dive today. We have over 600 people registered from 70 countries, and it's great to see you all joining us as we speak. So as Laura mentioned, this deep dive focuses on the Frontiers in Science lead article titled Enabling Sustainable Crop Protection with Induced Resistance in Plants, and it was published in October of 2024. Today's farming relies on two main ways to protect crops from pests and disease: using large amounts of pesticides and planting crops that have been bred to resist insects or pathogens. Neither strategy is sustainable. Pesticides damage the environment, they harm human health and contribute to global warming. Additionally, insects and microbes can evolve to beat both strategies, which means they won't work in the long term. So what if we could make the plants more resistant to pests and to these pathogens? A new technique called induced resistance in plants helps those plants fight pests and disease. It is also more sustainable than current methods. In their Frontiers in Science lead article that you see displayed on screen, the authors outline the many benefits of this more sustainable strategy for crop protection and how it can be adopted more widely. We are going to discuss today how further research, stronger regulatory support, and economic incentives are key next steps for the widespread adoption of induced resistant technologies. Around this lead article, we have built an article hub of complementary content to maximize its reach and impact, not only in the scientific community, but also among policy audiences and the public. So this is the article hub. It includes an editorial by Professor Nicole van Dam from the Leibniz Institute for Vegetable and Ornamental Crops in Germany, and viewpoint articles by Professor Jyoti Shah from the University of North Texas, who joins us today, and doctors Axel Youssef Mithofer and Alexander Furch from the Max Planck Institute and Friedrich Schiller University in Germany. We also have a policy outlook by Dr. Osama El-Lissi, formerly of the Food and Agriculture Organization in Italy. You can also find a lay explainer, some infographics, and a video, as well as an article in Frontiers in Young Minds, our journal for kids. So to start off today's agenda, we will begin with Professor Brigitte Mauch-Mani, who will set the scene for today's discussion. She will then be followed by her co-author, Professor Jurriaan Ton, who will discuss the versatile immune system of plants. Then Professor Tina Kyndt will discuss the insights from mode of action studies in induced resistance. And Professor Maria Pozo will discuss how microbes can boost plant immunity, leading to induced resistance. Following these presentations, we will have a panel discussion and a Q&A session with all of our speakers and today's guest panelist, Dr. Jyoti Shah, which is one of the contributors to the article hub. So let's turn our attention to our first speaker. Professor Brigitte Mauch-Mani is an emeritus professor at the University of Neuchâtel in Switzerland. Throughout her career, she has helped us understand how plants interact with microbes, how they defend themselves, and how they respond to stress. Her research has led to key discoveries, such as how grave pines fight downy mildew and the role of transcription factors in plant defense. She is also a specialty chief editor for Frontiers in Plant Science and a driving force in the field, with her work helping to develop sustainable agriculture, improve crops, and make food more secure. Brigitte, thank you very much for your leadership and in bringing this article to Frontiers in Science, and the floor is yours.
Hello, everybody. I will start with my first slide to dive in. So, as you might know, for at least 90% of its existence, humanity lived as hunter-gatherers and population densities stayed really low. Little groups of people were living together. But about 10,000 years ago, humans began to settle and formed larger communities. This period is known as the Neolithic Revolution you see right at the beginning. People started to live in towns. They didn't have this before, and population densities increased and occupational specialization ensued. So this means farmers were now responsible to feed the growing population. So we can say this is the moment agriculture, as we kind of know it, was born. In the 1960s, the Green Revolution led to a new era in agriculture. The introduction of hybrid crops, synthetic pesticides, fertilizers, special irrigation, and so on led to a boost in production, but sadly also to high social and environmental costs. So new ways were sought to protect plants and raise or at least maintain yields. Steps on this way, as you can see, are integrated pest management or new technologies such as the use of GMOs and genome editing. And as we believe, today the time has come for induced resistance. When we look at domestication of plants, we get new challenges. increase in human populations, as you see in the left, leads to larger arrays of the same crop being planted, and this makes plants more susceptible to pathogens and pest attacks. And this again, as you see on the right, to famine. The picture you see shows a scene in Ireland, and probably you've all heard of it, where around the middle of the 19th century, a period of mass starvation broke out due to almost total loss of the potato crop. This was because of an epidemic of diny mildew. It was really serious, and about 1 million people died at this time, and at least another million had to leave their homeland and were refugees in other countries. Globalization also favors new disease outbreaks. I remind you of the olive dieback, which was started several years ago. This was due to a bacterium, Xylella, which was introduced with ornamentals from the Americas into Europe, and it caused the death of many old and young olive trees. Another example, the fall armyworm, who attacks crops such as corn, sorghum, rice, cotton, wheat, you name it, it has many hosts. And it is also indigenous to the Americas, but by now it has spread to almost all African countries and many others around the world. So these are great problems and challenges we are facing. Besides globalization, I think food security is also threatened by climate change and stricter legislation concerning pesticides. If you watch the news on TV, for example, about every day you hear some problems we have with too much water, not enough water, too much heat, etc. So this is not going to go away by itself either. But as I hope you know, the world outside is still green. Maybe not in winter as now, but plants are still living. So this is good news. And this means that plants can defend themselves and adapt to these new situations I was just describing. And why can they do this? It is because they have an immune system. And part of this immune system can adapt to new situations, and this is what we call induced resistance. In the following, my colleague Jurriaan will introduce you to this immune system, as was mentioned before, and Tina and Maria will then go deeper into the subject and tell you how induced resistance can be switched on in plants and how it's supposed to work.
Thank you, Brigitte, for this important overview of the key messages of the lead article, and that sets the scene perfectly to now move on, as you said, to our next speaker and co-author, Jurriaan Ton, Professor of Plant Environmental Signaling at the University of Sheffield School of Biosciences in the UK. We'll now dive a little bit deeper into the versatile immune system of plants. Jurriaan is a preeminent plant pathologist driving the exploration of plant adaptation to recurrent stress from pests and diseases. His lab focuses on the molecular perception and signaling of chemicals that prime plant immunity, the so-called plant vaccines, and the functional relationship between root exudation chemistry and root-associated microbial communities to boost plant resilience. As an executive board member of the Sheffield Institute for Sustainable Food, Jurriaan collaborates closely with agricultural stakeholders to translate fundamental research on plant model systems into practical, sustainable solutions for crop protection. Jurriaan, thank you very much for being here, and the floor is yours.
Thank you very much, Rui. And it's a real honor to be here. Thank you for the invitation. And of course, a big thanks also to Brigitte to involve us in this endeavor. So yeah, as Rui said, I'd like to dive a bit deeper into the immune system of plants. And in the next 10 minutes or so, I hope to convince you that the plant immune system is not a monolithic on-off switch, but is much more sophisticated and versatile than that. And I like to do that on the basis of an illustration of this lovely tomato seedling. So when this seedling is exposed to attack by a pest or a disease, in this case a bacterial disease, the first layer of immunity it faces is innate immunity. Innate immunity operates according to a genetic program, and it hinges largely on surveying systems, as is shown here in this inset of this plant cell, and that can sense danger signals that indicate upcoming attack. Really important here are these cell surface receptors that stick their sensor domains into the intercellular space, and they can detect non-self signatures. These are chemicals that are unique to the attacker, or they can detect damaged cell signals, which indicate also attack by, for instance, caterpillars, for instance, breakdown products of the cell wall, or chemicals that should be inside the cell and start building up in the intercellular space. Now, if these things start to detect these danger signals, they initiate a rapid signal transduction pathway that leads to the activation of biochemical and mechanical defenses. And that is called pattern-triggered immunity. That's the default component of the plant's innate immune system. There is an extension to that as a consequence of an evolutionary arms race, very specialized pathogens, biotrophic pathogens. They employ virulence factors, we call them effectors. They're often injected into the cell, and as a consequence of co-evolution, plants or some plants can detect these effectors, and they do that by resistance proteins, or NLR proteins as we call them, that are encoded by resistance genes. And these form small lumps of resistosomes that then initiate a hypersensitive immune response. Now, both ETI and PTI can be very effective, and surprisingly, they act through similar pathways, and they also employ similar defense mechanisms. It's often the timing and intensity that is different. Now, I'm not going to go much into detail. I would like to flag up these two hormones that are really important in innate immunity and also induced resistance. Jasmonic acid is really important for the activation of defenses against herbivores and also against necrotrophic pathogens that kill the tissue and use dead cells to colonize their host. And then salicylic acid and its receptor MPR1, they are very important in activating defenses against biotrophic pathogens that rely on living cells. Okay, so as I said, innate immunity operates according to a pre-existing genetic blueprint. And that doesn't suggest much adaptiveness, as Brigitte also alluded to. And of course, plants are rooted to the ground. They kind of run away. They're also at the bottom of the food chain. So it's almost unavoidable that plants must be more adaptive to really cope with the constantly varying stresses in their environment. And this is where induced resistance comes into play. Induced resistance allows the plant to really fine-tune its innate immune system. And in many ways, it can be seen as the equivalent of the adaptive or acquired immune system in humans and other vertebrates. But of course, it relies on different mechanisms. Plants don't have B cells and they don't have T memory cells, and they use entirely different mechanisms to change and to elevate their resistance. So induced resistance can make genetically susceptible plants more resistant. How does it work? Well, it starts the same. It starts the same as in innate immunity. When plants are exposed to localized attack by either a pest or a herbivore, there are a multitude of long-distance signals that are produced, and they prime distal plant parts for the innate immune responses that are effective against these attackers. And that's what we call priming or sensitization. And this works both in response to pathogens, in which case we call it SAR, systemic acquired resistance. It also works in response to herbivores. Now that leaves the impression that induced resistance can only be triggered by hostile organisms, pests and diseases. Interestingly, that is not the case, because also beneficial microbes, particularly those that live below ground, can trigger an induced resistance response. For instance, plant growth promoting rhizobacteria that are recruited to the root surface and the rhizosphere, thin layer of soil surrounding the roots, but also fungi, some of which are endophytic, like mycorrhizal fungi. They're very good in triggering an induced resistance response that conceptually also relies on a priming of innate immunity. Now, not shown on this graph, but really recent evidence suggests that particular longer-lasting induced resistance relies on so-called epigenetic mechanisms that in some cases can even be passed on to the next generation. So induced resistance has both a physiological and an epigenetic basis, whereas innate immunity on the left-hand side there is genetically determined. Okay, now these are all internal strategies. Plants have more tricks up their sleeves. They can even cry out for help and recruit allies in their fights against pests and diseases. And these sort of ecological strategies, we refer to them as indirect induced defense. The classic example here comes from plants attacked by caterpillars that causes mechanical damage, and that releases these volatile organic compounds, these airborne signals, these odors that can be recognized by beneficial insects. On this cartoon here, you can see two parasitoids flying to the caterpillar. These cues can also be picked up by predatory insects like mites, and they attack the caterpillar, and by doing so, they protect the plant indirectly against herbivory. Hence the name indirect induced defense. Now, for a long time, it was thought that this indirect induced defense only works above ground with volatiles and only in response to herbivory. But recently, actually, we came to the conclusion, and by we, I mean the whole community, obviously, is that it actually also operates conceptually similar in response to disease. When plants are exposed to disease, either above or below ground, they're known to change their root exudation chemistry. And as a result of that, they recruit and select for different members in the microbial communities that are associated with their roots. We call that the rhizobiome. And these changes in the rhizobiome composition can result into a more disease suppressive activity by these bacterial and fungal communities. Now, one of the mechanisms by which these altered microbial communities protect plants against disease, I already introduced that in the middle, that is ISR-induced systemic resistance. So it's a multitude of mechanisms that are responsible for the protection there. Now, and then to make it even more complex, biology is never simple and straightforward. The volatiles that I mentioned before that can be triggered by pathogens, but also in particular by chewing herbivores, they not only recruit natural enemies of the herbivore, but they can also prime distal plant parts and even neighboring plants. And in that case, you call that VOCIR, volatile organic compound induced resistance. And in popular terms, this is often referred to as plant-plant communication. So this was a short whistle-stop tour, and I think it shows very clearly that the immune system of plants is not monolithic. It is not a binary on-off switch. It's much more sophisticated than that. It has different layers. It operates over different and expanding timescales. And these mechanisms also interact with each other. So the next interesting challenge then is how can we exploit it? And I will be very brief here. But of course, we have been exploiting the innate immunity for quite a long while already. For decades, crop breeding companies have worked with scientists to select for these R genes that provide effector-triggered immunity that are so effective against biotrophic pathogens. And with the latest advances in genetic modification, in particular CRISPR-Cas gene editing technologies that allow you to multiplex multiple genes in one plant, it is even possible to stack these R genes, and that makes ETI as a component of the innate immune system much more resilient against co-evolution. But of course, there are pathogens and pests that are not controlled by ETI. They're insensitive to effector-triggered immunity. They rely on PTI, and the genetic architecture of PTI is much more complex. But there too, there are opportunities, in particular the hidden genetic diversity in land races of crops and ancestors of crops, wild ancestors, can be exploited to identify genes, quantitative trait loci that make PTI more efficient. But what about induced resistance? Because this forum is about induced resistance after all. Well, there too, there are loads of opportunities. There are chemicals, we call them priming agents or plant vaccines, that are very good in mimicking biologically induced resistance responses. And they are particularly helpful for hydroponically grown plants because you can very precisely administer these chemicals without causing too much of an overstimulation that could go at the cost of plant growth and yield. But it can also be formulated and applied as a foliar spray or coated onto seeds. ISR eliciting microbes, there are success stories there as well, and Maria Pozo will give some examples about that later on. Particular fungi seem to work really well. Endophytic fungi, they can be formulated as a microbial inoculum and can protect plants via this ISR, this form of induced resistance by beneficial microbes. I also briefly alluded to the epigenetic basis of induced resistance, and Tina Kyndt will delve into much more detail about it. But this is a relatively new emerging area of research. My lab is working on this as well. And there are loads of opportunities, in my view, that can be exploited. So whereas innate immunity can be exploited by genetic modification, maybe we can exploit induced resistance through epigenetic modifications. And more about that by Tina later on. And then my last point is about the ecology of indirect induced defense. Well, there are already success stories there. Parasitoids and predatory insects that are already widely used for biocontrol, particularly in glasshouses where they can't run away. And also these microbial consortia that are shaped by stressed plants. There is a renewed interest, there's a real interest in these microbial communities. And in particular also in relation to the ISR eliciting microbes that I mentioned before, because it could be that a consortium might be more robust than a single isolate of a microbial inoculum, because you expect actually quite a lot of a single inoculum to perform well in different plant species and different agricultural ecosystems. So maybe the use of consortia is more effective. Now, if you go down that road, I would strongly suggest to do that in parallel with studying the genetic basis of root exudation chemistry, because the biology dictates us that it is the chemistry that shapes these disease-suppressive microbial consortia. So these consortia, these microbial, these disease-suppressive root microbiomes, and the genetics underpinning root exudation chemistry, they're two sides of the same coin in my view. And well, this is all from me for now, and I'll hand over to my colleague Tina. Thank you very much.
Thank you, Jurriaan, for this presentation. And as you said, biology might be difficult, but it can also be very interesting and cool. So thanks for this overview. And you know, just as a reminder for our audience online, if you have any questions about what's been presented, please make sure that you add them to our Q&A chat. And let's move on to our next speaker and co-author, Tina Kyndt, Associate Professor at Ghent University in Belgium, beautiful city. Tina heads the Kyndt lab, where her team specializes in plant immunity research, particularly against parasitic nematodes. and the role of epigenetics in rice stress responses. Tina and her team have also discovered and patented several novel biopesticide molecules that induce resistance to control nematode infections in crops. Their application could offer an effective and environmentally friendly alternatives to synthetic pesticides, enhancing sustainable farming practices. Tina will now discuss these insights from a mode of action studies on induced resistance. Thank you very much for being here, Tina. The floor is yours.
Thank you for the nice introduction. And I also would like to thank the Frontiers team for giving us this opportunity, for their excellent professional support, for the nice slides that they have created based on our input, and for their help when writing this review together with the co-authors. So my talk is about the mode of action of induced resistance, and this is based on insights, of course, from many plants. Initially, the field has focused a lot on Arabidopsis thaliana, as the model plant for dicot research. But more and more over the years, we see that many researchers in the field are going towards crop plants, dicots like tomato, for example, and monocots like rice, where my research is mainly focusing on. And there we have discovered that this innate immune system that Jurriaan has nicely introduced, but also the adaptive part of plant immunity is very conserved between monocots and dicots. And so it all contains many layers, of course, as Jurriaan also explained to you, and interaction. So it's a complex network that you can trigger from many sides. Now, if we focus on induced resistance now, and this slide does not change. Aha, yeah. Then we first see here in the blue line, you see what happens over time when you challenge a plant which has not been treated with a stimulus of induced resistance. When you challenge it with a pathogen, it can be a nematode, like the nematodes that I'm working on are little roundworms that are in the soil and that infest plant roots. If you challenge such a plant with that nematode, you will get a response of the plant. The plant will respond by activating some defense pathways. This is the normal state, let's say, in any field around the world. These plants will try to defend themselves. But when you have an IR-treated plant, you get an extra layer. When you have an IR-treated plant, you do a pre-treatment first. IR-treated plants, you treat them first with an IR stimulus early in time. It's typically a preventive treatment that you do. And upon a challenge, then the subsequent challenge by the pathogen, you get a stronger, a more robust, often also a faster defense response. So these plants are strengthened. They are better protected against the pathogen. And in that sense, we call this also, we refer to this as a similar phenomenon as vaccination in humans, something that we all know now. Since the corona pandemic, we don't have to explain vaccination anymore. Everyone knows what that refers to. So first you have this direct defense response, and only later you get this primed defense response, this stronger and more potent reaction to the infection, to the challenge. So that is why we sometimes call IR stimuli also plant vaccines. Now, which vaccines do we have for plants? Which stimuli can do this? What can trigger induced resistance? Now, over the years, a lot of research has, of course, been done in this field, and many different triggers have been identified. On the left side, we see the priming compounds, the priming agents, also sometimes called. These are typically natural compounds that are known to activate the immune system of the plant. For example, chito-oligosaccharides, oligogalacturonides, and a product that was made based on the combination of those two, COS-OGA, that you might know as commercialized as Phytosafe. Then we also have some hormone analogs that can trigger IR, some laminarin, which is an algal extract. BABA, a very well-known priming agent and very often studied in fundamental research on induced resistance. And then some things that have been more recently discovered, like, for example, the dihydroascorbic acid, which has been researched in my own lab and is the oxidized form of vitamin C. In the middle, we also see the microorganisms. A lot of microorganisms, beneficial microorganisms, can trigger induced resistance, such as the plant growth promoting rhizobacteria, the mycorrhizal fungi, and plant growth promoting fungi as well. And then very recently, we have discovered, based on the idea that, the mode of action studies that have been done in the lab of Jurriaan, for example, it has been discovered that epigenetics is a very important driver for the primed defense response. And that is why we can also apply this knowledge now and use inducible expression of a DNA demethylase, for example, to epigenetically modify the plant, and in that sense trigger induced resistance with recombinant DNA. So there are a few compounds that I wanted to highlight here that are already on the market. Just a few examples, like Phytosafe, which is the Cos-Oga combination, and Vaxiplant, which is laminarin, which contains laminarin as active substance. But also for the mycorrhizal fungi, we have Trichomic, for example, here in the middle, which is a combination of two species of mycorrhizal fungi from the genus Glomus and Bovaria. Now, what do we know about these mechanisms now? What happens in these plants? Well, by looking at the many research papers that have been published over the years in multiple species, we know that in general, when you activate AIR in a plant, you see the activation of PR genes, pathogenesis-related genes, encoded by encoding pathogenesis-related proteins, of course. Moreover, as Jurriaan already explained, you have the activation of hormones such as salicylic acid, jasmonic acid, but also ethylene and ABA can play an important role in induced resistance. The signaling molecules that are generally involved in the systemic spread of the signal are, for example, the ROS, reactive oxygen species like H2O2, and reactive nitrogen species like nitric oxide. Downstream of all these changes, of all these initial changes, you have the secondary metabolites that are being formed. These can be formed throughout the plants, both above ground and below ground, and typical examples of that are the phenolics, the alkaloids, and the diterpenoids. Now, of course, these secondary metabolites do vary between species, because between different crops, you will have other predominant secondary metabolites being formed, like in the monocots. For example, in rice, you have a lot of diterpenoids, while in tomato, you will have more alkaloids being produced. Now, this systemic signaling is not completely understood yet. We do know that there is some kind of communication between the foliar part of the plant and the roots, for example. That means that if you spray a stimulus on the leaves, you can get results in the roots. So we do see systemic effects, but also vice versa. If you have an AMF, artopuscular mycorrhizal fungal inoculation on the roots, you can see effects above ground. So there is two-way communication between roots and shoots that is not fully understood yet, and that certainly deserves further investigation. Moreover, because we activate all layers of plant immunity, we typically see a broad spectrum effect against multitude of pathogens and pests. One example that I wanted to highlight here of such a systemic IR stimulus that was discovered in my lab is dihydroascorbic acid. We see that it reduces, if you do a foliar spray, we see a reduction in the number of galls, the number of nematodes, and the reproductive stage of the nematode, egg-laying females. And those three parameters tell us that the plant is protected. There's around 30% reduction in all cases. And that is also something that I wanted to highlight here. The reduction potential that you have with induced resistance, it's never 100%. It's always a partial resistance that you get, around up to 50, some cases 70%, but let's say that 30% is already a quite strong effect, as we see here. When we did the DHA treatment, we were questioning how long does this work? Because when we do the treatment, that's a post-treatment, a foliar application, and we left different time intervals in one of our experiments between the treatment and the inoculation. And the slide that I showed you before was a time interval of 1 day. So if we wait 1 day between the DHA treatment and the inoculation of the roots, we see that all 3 parameters are nicely going down. We see a significant reduction in all cases. And here I have to highlight that 1 star means that the p-value is smaller than 0.05, 2 stars is smaller than 0.01. So it's strongly significant for the 2 last parameters. Now, if we waited longer, you will see that the effect diminishes. You see less and less effectivity of the treatment. So if you wait 3 days between treatment and inoculation, or 7 days, or up to 14 days waiting, it still works. But you see that the effect slows down, goes down. For example, at 14 days, only the last 2 parameters, the nematodes and the egg-laying females, are still significant, and the level of significance is lower. That tells us that you need a repeat. That you need, in this case of these post-treatments, you need to repeat your application. So multiple applications are needed. Again, referring back to the vaccination as if you do a booster treatment. So we recommend to do a booster treatment every 14 days to keep the defense memory awake. Now, if you then treat these plants for so many times with such an IR stimulus, you can question, doesn't that have a negative effect on the growth of the plant? And that is, of course, a very relevant question, because it is known that some IR stimuli, such as BTH, very well-known example of such negative effects of an IR stimulus on plant growth. And you see that in the top right corner here, a recent paper from 2019, where they see that if you repeatedly apply BTH onto apple plants, and they did the same in Arabidopsis in this paper, you see that the plants do not grow very well. So this growth defense trade-off is, of course, an issue in induced resistance that always needs to be taken into account and needs to be considered. So we decided to do the same for our DHA and check whether it would have negative effects on the plant growth. And we were very happy to see that it does not have this negative effect on the plant growth. DHA even slightly promotes the plant growth, and we get slightly more yield. It's not impressive, but at least the plants do not suffer as what you would get with BTH. So we now know that DHA is certainly not the exception. There are a lot of IR stimuli that can act both as induced resistance stimulus and as biostimulant. They can promote plant growth. Next to DHA, also mycorrhizal fungi, for example, have positive effects on plant growth. Now, what now happens in the next generation? There are a few pioneering studies that have investigated this in Arabidopsis. And in Arabidopsis, this was done already in the year 2012. This was published in Plant Physiology. And it is very clear that in the next generation, also these plants are protected. The effect is less strong than in the parental generation. You still see it, but the effect size is less, is smaller than in the parental plants. And also we see that the defense response is typically a bit more tailored. We know that this intergenerational effect that was published in the paper of Estrella Luna is dependent, for example, on NPR1, this important salicylate signaling molecule. We did the same in rice just to check, would this intergenerational effect also be seen in a monocot plant, because most of the initial research was done on Arabidopsis. And we did the treatment every 2 weeks with DHA. And to our happiness, we saw indeed that DHA also can trigger this intergenerational effects. So the descendants of the DHA-treated plants are also less susceptible to the nematodes. when we did an RNA sequencing analysis on the roots of these plants, we noticed that in those plants, defense genes were activated, and a subset of the genes that were activated of the parental plants were also found back in the daughter plants, showing that some memory of the treatment is passed down to the next generation. So we call this intergenerational acquired resistance. What underlies the intergenerational acquired resistance? Well, we do know now, based on an embryonic RNA-seq analysis, that this is related to changes to the epigenetic makeup of the plant. We see a lot of changes happening to the chromatin structure, to the histones, to DNA methyltransferases, and so on. So, and this is my last animation, epigenetics is an important driver, as Jurriaan also said, for the induced resistance phenotype, both within the same generation and over the generations, and is currently a field that needs a bit more exploration. With that, I want to thank you for your attention.
Thank you, Tina, both for the presentation and for the kind words at the beginning. And it has been a pleasure to bring this research to our audience, and I'm pretty sure they are all enjoying it so far. So our final speaker today is Professor Maria Pozo of the Spanish National Research Council. Maria is a biologist and scientific researcher with expertise in arbuscular mycorrhizal symbiosis and its effects on plant health and immunity. Her groundbreaking work exploring how beneficial soil microorganisms can be harnessed to improve plant health and productivity has advanced sustainable farming practices and enhanced crop resilience to climate change, all while reducing the need for chemical inputs. Maria will now discuss how microbes can boost plant immunity, leading to induced resistance. But first, I would like to take a second to congratulate Maria on becoming a professor in just very recently. Congratulations, Maria. Thank you very much for being here, and the floor is yours.
Thank you very much for the nice introduction and also for this nice surprise. So yeah, very happy about that. I will finish the session today focusing on the role of microbes in inducing resistance. And after the presentations we had before, we already, let me see, yes. We already have learned a lot on how the immune system of the plant have to be ready to detect aggressors and respond to them. And this immune system will activate defenses according to the type of aggressor and then stop the bad ones to come in. But then the plant also have to interact with many beneficial microbes. We all know many examples of beneficial microbes, and they are very common in the rhizosphere. So how the plant will detect or respond to these beneficial microbes, because they are also fungi, they are also bacterials, and they do have these patterns associated to fungi or to bacteria. So they are recognized by the immune system. And thus, the plant will respond and activate immune responses on the first time, right? But then there is a precise communication between the microbe and the plant, and there is a recognition that it is a beneficial. And this tune down the defenses, and even there is an activation in many cases of a symbiotic program that the plant actively welcome the colonization or the entrance of the microbe. And this is, for example, the case of mycorrhizal fungi. Are the ones which we work more, and we have here some examples. We can I don't know if you can follow this slide, but when the hyphae of the mycorrhizal fungus arrive to the root surface, there is recognition, there is some activation, there is a ROS production, nitric oxide, there are an exchange of effectors and mutual signals. But at the end, the signal signature is specific for beneficial, and the plant modulate the response to welcome and to actively promote the colonization. And from long, our idea is that this modulation of plant immunity during the colonization of the tissues by the beneficials leaves a memory of this initial stress or this training of the immune system, resulting in a prime defensive state in the plant, or an alarm state, so that the plant will be able to respond more efficiently to future challenges. And again, we come back with the concept we have been looking before to this vaccine effect. So I just want to show some examples on how we know that microbes, and in this case, mycorrhizal colonization of roots, does change the regulation at the transcriptional level. Also, many secondary metabolites are altered, and the homeostasis, the hormonal homeostasis change, and this makes the plant more efficient in responding to different challenges, not only in the roots where the colonization is taking place, but also in the distant part, as we saw from Tina on the other way around. They were applying above ground, and you could see the activation of defenses or this priming effect also in roots. And now we can see that mycorrhizal colonization can let the plant more resistant to different aggressors above ground, necrotrophic pathogens and herbivores. So this enhanced resistance is taking place through different mechanisms, and one of them is this enhanced capacity to promote structural barriers. So a control plant will also respond to a pathogen trying to penetrate the leaves, as we see in the upper part, by the accumulation of callose. So cell wall compounds will make a physical barrier to prevent the fungus to penetrate. But then this accumulation of callose is stronger in the plants that are elicited with, in this case, with mycorrhiza, but with IR. And we see that this is similar to what is happening also in roots. We have in the bottom part, tomato roots responding to the Oomycete Phytophthora parasitica. And again, the hyphae of the Oomycete is trying to penetrate in the cells, and there is accumulation of encasement material to prevent forming a papilla to prevent the fungus to go in. So there are stronger structural barriers that are related to a more efficient activation or mobilization of resources. So somehow the induced plant is able to reallocate and to redirect the resources more efficiently. So it's not all about physical barriers, but also chemical barriers, and there is a higher accumulation of defensive compounds in response to pathogens, but also in response to herbivores. And I'm showing here some examples for Dr. Siwa and Tuta absoluta, as they are both Lepidoptera that we are studying in our lab, but especially I want to focus on Tuta absoluta, as it is a major problem in tomato production around the world. And just as an example, using untargeted metabolomics, we can see how this control plant, when challenged with the herbivore, I label with an H there is sometimes some small accumulation of some defensive compounds, or here is not even significant, but then is the mycorrhizal plant that without herbivory, the levels are similar to the control or to the non-induced plant. But then upon herbivory, there is a strong accumulation of this compound. So the priming response that we just saw in the scheme from that Tina presented us. And well, we are trying to focus on these compounds with differential accumulation profiles, and looking for the ones that are accumulated to a higher extent in the more resistant plants. And when they are available commercially, we try them to see to functionally characterize them, and we could confirm that they have insecticidal properties. So they have anti-herbivory properties, showing again that then this differential or this stronger activation of defenses is efficient in containing the aggressor. Then, of course, we are interested in knowing how is this regulated, how it comes that with the same attack, then the induced plant will be able to activate more efficiently the responses. And then, thanks to the collaboration with the group of Kristina Gruden in Slovenia, and using this mechanistic plant stress signaling model, we went through a transcriptomic analysis of tomato plant responses to this leaf miner, the Tuta absoluta, and through time we can see how the main nodes of regulators of the signaling pathways involved in the regulation of herbivory responses, like mainly jasmonic acid and ethylene, they do appear in both in non-mycorrhizal and in mycorrhizal plants, but we can see more nodes and more connections in the mycorrhizal plants. So it really seems that so the pathways are the same, but the induced plants can shape or fine-tune more efficiently the coordination of the responses. And we do think that this is the reason why, upon attack, they can use more efficiently the resources. So we wanted to know if we can see that and if we can apply that really, I mean, to real agronomic practices, and in fact to modern practices where you have enough nutrients and good conditions for the plants. So to test that, we use different fungal strains to inoculate seeds at the nursery stage. So we do vaccine the plants before we bring them to the field. And thanks to collaboration with growers cooperatives, both in open field conditions like more traditional tomato growing or in greenhouse like more protected crops, if we could see the effect under these conditions. And in both natural enemies for biocontrol of pests was used. So already using integrated pest management, especially in the greenhouse conditions. So what we saw is that indeed there was a reduction of Tuta absoluta natural incidence, not only in leaves, but also in fruits. We have here just in blue and orange, just two different mycorrhizal fungal strains, and they both efficiently reduce the incidence of Tuta. And it's not only about this specific pest, but we saw in general a better sanitary status as other attackers were also showing less incidence in these plants. And in some of the treatments, we found a yield increase up to 15% in the final production. So that was really good news, because we, first, because of the cost of induced resistance, that sometimes is a question to address, as Tina just pointed out, but also because in these conditions is high fertilization is used. So still we thought maybe we will not see the benefits of mycorrhiza there, but we think that the yield increase is mainly associated to less cost of the stresses in general. And in addition, we saw that, and other reports also show that there is an improved nutraceutical value of the fruit. So there is this accumulation of secondary metabolites in leaves, but also in fruits, indeed give an added value to the fruits, including some increases in carotenes and lycopene. So we do have an improved or a stronger direct defenses. We do have other additional benefits, but we also thought or observed that the protection in the field was even stronger than we thought it will be, just because of increasing direct defenses. And then we tried the other, or we addressed the other level of protection that Jurriaan introduced in his talk on the contribution of indirect defenses. So when the plant is attacked, we just saw that there will be changes in these volatile organic compounds, and they will attract natural enemies of the pest, predators and parasitoids. And what we observe is that when the plant is already induced, these changes in the volatile blend are more profound. And if we did some bioassays with choice test between non-mycorrhizal and mycorrhizal plants, and when they are challenged with the pest, and we release the predator Nesidiocoris tenuis that is commonly used in for example, in southern Spain, in most greenhouses to control pests, it does prefer to go through to mycorrhizal plants. So there is a synergistic effect or a stronger call for these natural enemies. So if we look at what we have been listening today, induced resistance have a wide efficiency spectrum. So that is good news. It's long-term protection. In the case of, I just showed from the greenhouses and the open field, we just followed the across the production season, so till the end, and we still saw the effects at the end. It is compatible with natural enemies, and not only compatible, at least in the systems we have tested, there is even a synergistic effect. There can be higher yields, there is improved nutritional value, or can be improved nutritional value. And I want to put emphasis on the idea that because of the induced resistance is resulting from different mechanisms. We just heard stronger structural barriers, stronger chemical weapons, and also stronger indirect defenses. Then there will be a lower risk for pests to develop resistance. And I think that is really very important. And what we want to point now, then, according to this, is that then induced resistance can complement very nicely the integrated pest management programs. And I think we need to do a stronger effort on considering induced resistance in this whole view of integrated pest management. So as a summary of the session today, I think we want to point out that the induced resistance by itself cannot solve all the problems of modern agriculture. And as Tina mentioned, we will not get 100% protection. It's not that we make completely resistant the plants, but they will help to reduce the excessive use of pesticides. There is already a significant reduction of damages and of the progression of the attackers. They offer long-lasting protection, and they help to improve the nutritional quality and health benefits of crop products and, of course, of the environment. So we believe that the induced resistance used in combination with other sustainable methods will prove to be an effective approach to pest and disease management and promote biodiversity and soil health. And we need to conserve biodiversity, and I think soil health has been forgotten for long in agriculture, and this is now a key point for future steps. And with this, I think I'm finishing and giving word for the panel.
Thank you, Maria, and for this overview and for this important point about soil health being really vital that we pay attention to it. And I can see already some questions from your presentation coming in, so hopefully we'll be able to address some of them in the Q&A session. And thank you to all of our speakers once again for all of your insights for today's discussion and setting the scene perfectly for our discussion panel. So the panel discussion provides a great opportunity to expand and dive deeper into the issues and themes that were covered by our speakers. During the panel, we will also be taking questions from the audience, so please make sure that you leave your questions and to which speaker you would like them to be directed in the Q&A chat. We also have a guest panelist joining us today, so I'm delighted to welcome Dr. Jyoti Shah. Dr. Shah is a distinguished research professor and chair of biological sciences at the University of North Texas in the US. Over the past 25 years, he has led a distinguished career in plant immunity, developing an international recognized program focused on plant defense signaling. His work has also led to new ways of reducing fusarium head blight disease in wheat, and he has also developed key tools to study plant defense against aphids. His contributions have significantly advanced the understanding of plant pathogen interactions and also contributed to the impact in agricultural practices globally. Welcome, Jyoti, and thank you for being part of this event today. And so to begin the panel discussion, Jyoti, I would like to turn to you first, if that's okay. And I have a question related to your viewpoint article that you contributed to in the article hub. But first, I would like to ask you if you would like to share some of our reflections of this Frontiers in Science article and what has been presented so far today.
Thank you, Rui, for the wonderful introduction, and thank you for to the organizers for organizing this deep dive, and of course to all the authors of the paper, which was very timely, I think, insightful in providing us with the history of IR, the mechanism underlying it, its importance, but at the same time the challenges with its application, and proposing some, some ideas about how solutions could be found. What I want to add to that again is that just a point I want to make is that while we think about these technological developments, application of IRs, we need to not forget that 80% of the farmers are poor farmers. They're not in, and I think whatever technology we develop, can it be useful for them? And what I see a positive here is the epigenetic effects of it, the next generation effects of ISR, which in countries where farmers retain their seeds, okay, that could be very beneficial to them. So I think I would, kind of want to highlight that part of it, when we're thinking of sustainable agriculture, but the wider application of sustainable agriculture. As far as just summarizing the presentations today, I think we started off with Brigitte giving us an excellent overview of, plant defenses itself and how what ISR is, but at the same time giving a historical perspective of agriculture itself. How over time technology has developed, it's helped agriculture, and now we are at a stage where IR has the potential to increase resistance and contribute to sustainable agriculture. That was followed by Jurriaan, who did a wonderful job of kind of introducing us to plant defenses. Again, plants being able to defend themselves is something which is very important, which as a teacher, I find many people don't realize it. Students don't realize it. So I think that's where this frontiers concept of getting students, kids involved in understanding it is great. That's how we can build up that kind of knowledge base. And when it comes to consumers, these guys will be the consumers in the future application of these technologies. So I think that was brilliant to have that. But at the same time, Jurriaan did an excellent job of explaining all the different mechanisms of defenses that plants utilize, their potential applications. He made it very clear how IR as well as indirect defenses have such an important role in controlling diseases and insect infestation, and how they can make, and this is very important, how they can make plants which are genetically susceptible more resistant, right? So that's a complementary mechanism, complementary to other strategies like R genes, as well as using pesticides and so on. And then Tina went on to talk about the mode of action of IR, both the physiological basis of it and the epigenetic basis of it. More importantly, the priming effects of IR and how they help plants to respond more robustly upon infection. She showed nice data with dehydroascorbic acid as to how it primes defenses, but does not have that cost of reduced yield, at least in her case in rice. And that's something which needs to be tested in different crops. Again, different crops, different varieties might behave differently. So that's something we should keep in mind. But again, it's very promising. And then, about microbes themselves being potential IR inducers itself, I think was something she introduced, which Maria jumped into in her talk, or brought up into her talk, about how mycorrhizal infection, colonization, I should say, can stimulate IR and provide broad spectrum resistance, not just in the roots, but also in the shoots, and also at the same time improve the quality of the product. And she showed nice data about how this could be applied in the field situation too, because in the end, that's what's more important is, can this be applied into the field situation? Is it agriculturally relevant? And can it work under different conditions? That's something needs to be tested. So I think a lot of potential here, I think, and it was great to hear all these presentations.
Thank you, Jyoti. And this is great, summary already, and I see some agreement and from the speakers, and so I think there's going to be a chance also to for them to speak on some of the points that you made. But before, I really want to also to go into the question that we actually had for you that after this summary, and it's like, you know, the lead article highlights that induced resistance is rarely used in agriculture production systems, despite its long history of research and known benefits. And I think you also kind of like addressed lightly this part, but you know, given your pioneering work in plant defense, signal and mitigation strategies, how can we best translate the research findings we have heard about today into practical applications, especially for the farmers that you mentioned? You know, because the most of the farmers are like you know, not companies that are working on, but day-to-day farmers that they're basically having their yields produced.
So I would start up by saying that, you know, when we think of agriculture, we need to think of farmer, but we also need to think the end, which is the consumer. Okay, the consumer demands what they want. Okay, and if the consumer demands certain things, then the baker or the breeder, and the farmer will grow that material, right? So I think this is, if we keep the chain in mind, and now we think of the farmer, you know, whatever strategies we develop, it's very important for us to communicate that, bring those stakeholders on board. So, you know, a discussion where we bring them all together, let them see it for themselves, how this is benefiting. When you think of a farmer, to farmers, something that they see is that, hey, I apply a chemical, it immediately controls the disease within a few days or so. They see the immediate benefit. If it's something which is going to be slow, they may not see the benefits right away. But if we bring them to the field, show them beforehand, this is how it works, this is how it's going to benefit, this is the cost effect to you, or cost benefit to you. Your yields are going to go by so much because you're taking this early step of plant protection or inducing defenses. So I think that kind of communication, education is very important. And I say this because I, you know, work on wheat head blight, and we meet every year. Okay, it's a small forum where farmers come in, the breeders come in, the millers come in, the other industry people come in, and policy managers, USDA and other people come in, and the research come in. We share data, we exchange ideas about what challenges they're facing. And at the same time, we also have people who are predictors, you know, who they are prediction models. When the disease pressure is highest, what should the farmers do? They communicate that live to the farmers. Hey, disease pressure in this area is high. You need to be doing this. You need to be planting these kind of varieties. So I think it's a combination of factors which come together: communication, timely communication, and education by people actually seeing, you know, for themselves the benefits. That would be the way to go, I think, you know.
Thank you, Jyoti, and this fantastic overview and for the answer. And you know, I'll give the chance also to the speakers. I know that it seems like some of you want to pitch in, so I'll you know, feel free to unmute yourself and just add anything to what Jyoti just said. And if you want to expand or comment any of the points, Jurriaan, I'll go to you first because you look like you wanted to say something. So yeah, but go ahead.
I always look as if I want to say something, but I thank you very much, Jyoti. That was a really nice perspective. And I want to come back to the first opportunity you mentioned, and it actually relates to the last point you made as well, that we need to reach out and engage with the producers of food, farmers. And also we need to realize that the majority of food on this planet is produced by subsistence farmers. And you made this interesting point in the beginning that, well, it is now emerging that there is an epigenetic basis to induce resistance, that induced resistance can be passed on from diseased plants to the next generation. Is it perhaps worth for the subsistence farmers to start collecting seeds from diseased plants? Now, I'm sure that Tina has a opinion here as well, but this is where the communication between scientists and stakeholders, farmers, is so important. First of all, of course, there are huge risks there because many diseases can also be transmitted through the seed, right? So you don't want to inoculate your next yield with your pathogen through seed transmission. But I'm sure that there are opportunities there for improvement. But we also need to be cautious that we're not over-egging these effects. The heritable induced resistance responses, and Tina said that as well, they are very subtle and they're also context dependent. And that's why we need to understand them better. We need to understand the costs, and we need to understand how these epigenetic modifications prime the immune system before they can be truly safely applied. I think that level of research is still at technology readiness level 1 or 2. But that wouldn't, stop us from, trying to move on in this innovation pipeline, I would say. Anyway, I'll stop talking now.
Thank you.
Can I
Yeah,
just-
Jyoti, go ahead, and then we can
I-
segue to Tina and Maria. I think also wanted to say something. Yeah, go ahead.
Yeah, I just agree with you, Ryan, and I think my point here is that, this has the potential, but at the same time, researchers should be engaged with those people in those countries too, because it's, there are other diseases there at the same time. What's the effect of one on the resistance to one on the other? Those need to be part of it. So it's not that education has to happen there as well at the same time.
Yeah.
Great, thank you, Jyoti. Tina, and Maria, do you wanted to add something to this point, this part of the discussion? Brigitte, also, please feel free to unmute.
I think indeed that I agree with Jurriaan that it is very that it's certainly not a good idea to exploit these intergenerational effects with infected plants. Collecting the seeds of infested plants also indeed in the next generation you will get less infection. That we have shown for in research. But what would be possible is that you treat your parental plants with a priming agent such as the DHA or a BABA, and then collect those seeds, and then you have the benefit that those descendants are again slightly protected, not as strong as the parental plants, but at least they have, this is possible, but you have to be certain indeed that your product that you apply in the first generation is safe and does not have negative effects on the growth.
Thank you, Tina.
Oh, sorry, my video was off apparently. I didn't notice.
No worries, no worries. We heard you just fine, so thanks a lot for that. Maria, do you also add something to add to this? Brigitte?
Well,
Maria? Yep.
I think Brigitte on the epigenetics, right? Brigitte?
Yeah, you can
Yeah,
go ahead,
cool.
Maria, and then we finish with Brigitte, and then we have a
Okay.
quick round of
Let's go on.
questions from the audience.
Okay, no, I was not adding on epigenetics. It's Jurriaan and Tina who are really into that. In fact, we know very little on the effect of beneficial microbes at that level. So I'm really looking forward what is coming next in the coming years. There is a lot more to know. And as Jyoti mentioned, we really need to know that more, but also like talking to the growers and also learning from their experience. So that's key. I just wanted to make the point of what you mentioned, that not only, it's not only one pathogen or one pest, and it's like agriculture is facing many different stresses, and mostly, and especially in those not highly technified areas, right? So I think this idea or an additional benefit of IR that we maybe didn't focus too much is that this general idea of responding better to the challenges, so mobilizing better your resources, it gives not 100% resistant to one pest or to one thing, but overall is helping you to react more efficiently to different things. So summing up, is not only one pest, or it's not only even a biotic stress. We know that when the plant is prime, it can also, because of this better orchestration of hormone homeostasis, I think they can respond better. And again, it's not a miracle. You don't get 100% out of whatever stress, but in general, it pays off. And we really to look at, as Jurriaan pointed, this context dependency. We need to know more what factors are influencing the induction, how we can optimize. And I think there are many things we need to look at, like fertilization and managing of the soil or techniques in the crops. If we know more, we will be able to tailor our approaches to promote or to get the maximum out of IR. So I really think that now that we are aware, and anyway, many pesticides are being banned, so we have to look for these other solutions. I think we will really get to see a lot of progress in the coming years.
Thank you,
And
Maria.
with that,
Yeah.
I want to give to Brigitte that.
Yeah, if it's okay, like I'll like to move on to Brigitte, because we're also approaching the end of the webinar, and yeah, I wanted to give the chance to Brigitte to say a few words and a few thoughts. Brigitte.
I agree with my colleague said, and I think one important point, not a scientific one, but it's to make really sure that you do not harm the plants when you do something to them, because once you do it, nobody will have confidence in you, and you couldn't go on working on this. Really make sure do not harm the plants.
Great, thank you. And if I can just have a few very quick questions to Brigitte, I would like to start with the one from Ramon Guevara Gonzalez, and he is asking, how might the induced resistance be introduced to the market, both as a service and as a product, and do you see this coming soon, or kind of like do you have an estimated timeline of how feasible this is?
I have a hope that it comes soon, but I'm not so sure, having hard times now, that it will be that soon. But we need help from governmental agencies and others to help introduce this to the farmers. It's not scientists directly who can do this. We need some communicators.
Thank you. Jurriaan, I have a question for you. This is from Leslie Weston, and she's interested in the duration and persistence of induced resistance. Can you elaborate on that?
Yeah, sure. Well, let's start with the persistence, and I think that has been for a very long time a underexplored area, because people always want to study the early events, and it wasn't until recently that we became interested in the durability of induced resistance, and this really led to the discovery that induced resistance can be maintained through the life cycle of the plant, and in some cases even be transmitted to the next generation. But in the absence of stress, it goes away. This is one of the reasons why we and others, that we started to consider epigenetic mechanisms, because epigenetic mechanisms enable the plant to do that, to maintain for extended periods a certain state, in this case induced resistance, but still be able to revert it, because we know that there are costs associated with induced resistance, whether we like it or not. And for a plant, it is, of course, an excellent mechanism to switch back to a different state that is not associated with induced resistance, but is less costly. Now, in terms of exploitation, and I'm now talking about the epigenetic component of induced resistance, I think the opportunities there lie mostly in understanding how the epigenome orchestrates the immune system, and then using untargeted and targeted pathways to manipulate the epigenome and select for lines that are innately epigenetically primed. And I think that is a very promising way forward. But as Brigitte said, it will take a long time. These sort of applications take time to really develop. Yeah.
Great, thank you. Maria, I have a question from you from Nazib Zaman, and I also a little bit curious about this. Can we protect crops from weeds by using chemicals that are secreted by fungi?
Oh, yeah, that's another topic. It's not induced resistance, but then there are many. So I think we really are underexploiting all the potential of microbes, and especially fungi are magnificent factories of compounds, and they're far to know. And of course, there is a lot much more known like in biomedicine, and like we know there are many compounds acting as medicines, and we really need to explore more on our soils, and that's why we have to keep biodiversity and then not using too much pesticides. But there are many fungi producing active compounds, and some of them will be need to be exploited, but we need to know more on what it will be better using the fungus, or in which conditions will produce the compound, or should we just work on getting the products out of culturing the microbes. So a lot to do again here.
Thank you, Maria, and thank you, Nazib, for giving us a potential new idea for another lead article for Fruities in Science. Thank you a lot for that. So my next question is for Tina, and it's from Karan. What are the potential challenges in balancing the activation of induced resistance in plants without compromising their growth or yield? And how can researchers ensure these technologies remain accessible and cost-effective for the smallholder farmers in developing regions, for example?
Yes, very good question. I think the main determinant factors for the balance between defense and growth, this trade-off effect that you sometimes see, are the dose, the amount of product that you apply to induce resistance. So if you go too high, you get into negative effects on plant growth. Like, for example, with the BTH, that were 6 applications that were shown there on that slide that I presented. That's a lot. So that's a very high dose, 6 applications of 1 millimolar, that's high. And then the plants start to suffer. So you have to keep your dose up to the minimum where you get the effect, but don't overstress the plants. And the second thing is the compound. There are compounds that simply are having this negative effects on growth. And BTH is, again, I'm sorry to say, the negative example there. And that's simply because it's a salicylate homolog. It's an homolog of a hormone. So you're kind of disturbing the internal hormone homeostasis of the plant. And you have to avoid that. So for DHA, for example, we don't see that, and that's probably because it doesn't affect this homeostasis. And in fact, we see even some growth promoting effects like with the mycorrhiza. So selecting the right trigger for IR and the right dose, those are the things that I would recommend.
Thank you, Tina. And I know we are approaching the end of the webinar, but this is such a fascinating discussion that if it's okay, I would ask for us to stay 10 minutes more so that we can ask a few more questions, and we can from the audience. And this one, it's directed to Jurriaan first, but then any other speakers are welcome to pitch in. And it comes from Isabel Cortez. So what are the causes, what causes the significant differences in results when using elicitors and biofungicides in lab versus field experiments? Do you think that all the experiments should be developed according to the hosts, the pathogen, the climate conditions, or a combination of these factors? I kind of have a feeling of the answer, but yeah, go ahead, Jurriaan, and then any other speakers that want to jump in.
Wow, that's quite a question. I think that induced resistance is context dependent, and it is important before large-scale exploitation to test the reproducibility, the potential costs, and the effect size under different conditions, under different growing conditions. So there's no doubt about that, because, and there's a scientific explanation for that as well, that induced resistance is a form of acquired immunity, as we all alluded to in our presentations, but it is deeply integrated in the same pathways that control innate immunity and even development. So if you pull one string in the signaling web, there will always be a ripple effect somewhere else. So I would strongly applaud the sort of approaches that both Tina and Maria were outlining, that doing field trials and see how these inoculums and these chemicals, these priming chemicals, perform under semi-commercial, commercial, or field conditions. And that will teach us actually quite a lot. I don't know whether that exactly answered your question, but I hope I at least went some way in doing that. Yeah.
Thank you, Jurriaan. I don't know if anybody else wants to jump in into around the question. No. All good. Okay. I'll go then to the next question to Jyoti, and and it's from Abdur Ramin. I hope I'm saying the name correctly. What do you think are the role that allelopathy plays in induced resistance and other crop resistant mechanisms?
I can say I'm not an expert at allelopathy, but if somebody else wants to take that question, that's perfect. But people have seen scientists have seen the effects of allelopathy in controlling, for example, controlling seeds growing around them or germinating around them. So I think it does have a strong potential, but again, it depends on what kind of plants you're looking at, what kind of chemicals are being produced, and what effects they have, not just neighboring plants, but the type of plants you are studying or you want to cultivate. So I think, again, I'm not the best person to answer that question, but it has a lot of potential. Maybe another topic for Frontiers to think about.
Yeah. Thank you, Jyoti. I believe that Jurriaan wanted to say something. Yeah, go ahead.
Yeah, it's a very good question, actually. And this is something that I am at the moment investigating with my colleague, Stephen Rolfe here at the University of Sheffield, that allelopathic compounds that are produced by plants when they are competing with each other and that are known to have allelopathic effects on neighboring plants of another species, they're often the same chemicals that I also refer to in my cartoon that can shape these disease suppressive microbes. So there is another cost, right? So on the one hand, there's a benefit that these chemicals can select and recruit for ISR eliciting bacteria. But at the other hand, too much of these chemicals can have a lingering effect and could actually really repress growth of crops. So you need to get the balance right and you need to understand the biology. But yes, allelochemicals is one word. You could also say semiochemicals. These are secondary metabolites that have multifunctional signaling functions in the soil. And it's really important for us to understand these better. But it's a very good question. Yeah, thank you.
Thank you, Jurriaan, for the answer. And this was a great discussion and a very interactive webinar. And I think that we might need to pitch to the comms team an extension, a special podcast, to go through all of the questions that we had from the audience. And I really want to thank the audience for the questions they are sending our way. But we are almost running out of time so for today's discussion. So. But before we close, I would like to thank all of today's speakers and expert contributors for their knowledge and for their contributions. Thank you very much to Professor Brigitte Mauch-Mani, Professor Jurriaan Ton, Professor Tina Kyndt, Professor Maria Pozo, and Dr. Jyoti Shah. Thank you very much for your insights. And I would also like to thank each and every one of you that join us today for this deep dive. I really hope you enjoy the session, at least as much as I did. Allow me to also share some of the information about how we can keep up this conversation going and let you know about some of our upcoming events. Please make sure that you visit the Frontiers in Science website to read the article we discussed today and its related content. You can continue the conversation on social media using the hashtag Frontiers Forum. Be sure to subscribe to Frontiers in Science updates, and you make sure to visit also the Frontiers Forum website for upcoming events. And with this, I would like to thank you also once again for now from me, for our guests and the entire team at Frontiers. Thank you, goodbye, and happy holidays.